Thank you very much. First of all, thank you very much to the organizers for inviting me. So I'm going to try to keep this talk short since we have lunch afterwards. So that's a good idea. Then, as you said, I'm going to talk about derivations of non-local PDEs for individual and collective movement, collective migration. And this is work together with few people that they used to be in Edinburgh at the time I did my PhD, and now they are all over the place. Okay, so my talk will be divided mainly into two parts. So in the first part, I'm going to clarify the form of biologically relevant PDEs for the non-local movement of individuals. In particular, I'm going to focus on the case of the bacteria E. coli. And this non-local movement will be described by fractional differential operators, which of course I'm going to introduce later on. And also the type of equations that I'm going to derive are going to be fractional chemotactic equations. So it's kind of related to Tommaso's previous talk. In the second part of my talk, I'm going to consider, well, basically I'm going to extend this non-local individual movement to a system of interacting particles. Okay, in this system, we are not going to only look at the non-local diffusion, but also to alignment and interaction between individuals. In particular, we are going to study two different limits, so the long-time behavior of this system and also short-time behavior. And all the results I'm going to present here are inspired by some biological evidence, which I'm also going to discuss later on. Please interrupt me at any time if you have any questions, okay? So before starting, I'm going to give like a brief overview of what do I mean by superdiffusion and what is the relationship with the fractional Laplacian. Okay, so let's start from the simple. So as we all know, the diffusion process is described at the individual level by a Brownian motion. Okay, and one of the main characteristics of this movement is that the mean square displacement grows linearly with respect to time. Then the distribution of the displacement of this particle follows a Gaussian distribution. Okay, and then in the limit, when we look at this process at the macroscopic level, this Brownian motion is described by a classical Laplace operator. On the other hand, for the case of the superdiffusion, it is described at the particle level by a Levy motion, the so-called Levy motion. And in this case, the mean square displacement grows non-linearly with respect to time, where alpha is a parameter that is between 1 and 2. And note that for alpha equals to 2, we recover the classical diffusion case, while for alpha equals to 1, what we have is ballistic motion. In this case, the distribution of the displacement follows a power law, so a heavy tail distribution. And as we can compare in these two cases, here we have a strong presence of long jumps. Okay, this gives the non-locality of the movement that I'm going to study. Of course, when I'm going to talk about cells, I don't mean that my cells are jumping. It just means that they are moving in the same direction for longer times. Okay, that's what I mean by a long jump. And as we will see in this case, in the superdiffusive case, this non-local movement will be described by fractional differential operators. Okay, so let's see what is the connection between these long jumps and the fractional operator. And this is an explanation that I always like because I think it's very, very intuitive. Also, it's very hand-wavy. Okay, no rigorous at all. But I think it gives a nice intuition behind this operator. So let's think about a very simple random walk. So we have a particle at position 0, and this particle can jump to the left with probability 1/2 and to the right with probability 1/2. So it's just jumping to the neighboring sides. Then the probability of finding this particle at position x at time t plus dt is given by this expression. So this is very classical, right? We all know this. Then we can rewrite this expression just by doing some very simple manipulations. We write it in terms of difference quotients, right? And then in the limit, when the x and dt goes to 0, we see that this jumping process tends to a diffusion equation, okay? So a classical heat equation. But now what happens if we allow this particle to give very long jumps? So my particle is not going to jump to the neighboring site, but it's going to give a jump of length d. In this case, the probability of a jump of length d will decay algebraically with this distance. And we do the same manipulations as before, so we can write this expression here, where of course we are not going to have 1/2 anymore, we have some other probability. And well, doing some very simple rescaling, let's say, what we observe is that when dt and dy goes to 0, just in this limit, very similar to the random walk that I explained before, we see that this right-hand side here becomes a Riemann integral, right? And this is how I define this fractional Laplacian operator. It's defined here. So this right-hand side in the limit can be written as this integral with respect to a non-local kernel. So here C alpha n is just a constant, PV represents the Cauchy principal value of this integral, and well, this is one of the type of fractional operators that you can encounter in life, let's say. I have to say this is not the one I'm going to use, but this is the most classical one, okay? And just by looking at the definition, you see already the non-locality of this operator, right? It's an integral in Rn with respect to a singular kernel, so you get the non-local movement. Okay, so with this brief introduction of what do I mean by superdiffusion, it's just a non-local movement, and this fractional Laplacian, how it is connected to this non-local movement. Then I'm going to start with the individual description, okay? So just the first part. Well, I guess we all know what chemotaxis is, but just for the ones that are not very familiar, just chemotaxis describes the bias movement of individuals in response to a chemical gradient in the medium. Okay, and in the classical case of chemotaxis, we have a particle, it's going to run for some time tau, it's going to stop, tumble, and choose a new direction at random. And this is also called the run and tumble movement, so I'm going to use this terminology. One of the main characteristics of this chemotaxis movement is that the runtime tau follows a Poisson process. Then, at the microscopic level, this chemotaxis behavior can be described by the classical Patlak-Keller-Segel equations, which are written here. So here, U, it's a microscopic density of the population. Rho is the concentration of the chemical in the medium. Then the first term here just describes the random component of the movement, while the second term just describes the bias of the particles towards high concentrations of the chemical. The second equation here is just describing the evolution of the chemical concentration, right? And for the sake of simplicity, I'm not going to consider that this chemical is consumed or produced by the cells. Okay, it's just uniformly distributed in the medium. But what has been observed in recent experimental results is that when the concentration of the chemical is sparse or absent in the medium, then the individuals change the search strategy, and instead of doing a very localized search, what they do is they keep moving in the same direction for longer times, describing this type of non-local diffusion. Okay, so basically here the main change is in the runtime tau. Okay, that's why in the first part of the talk, my main assumption is going to be that the runtime tau is distributed according to a power law. It's a heavy tail distribution, which basically means that the probability of having a very long run is non-negligible. Okay? And then at the macroscopic level, this non-local movement will be described by non-local fractional differential operators, as is written in here. Okay? So, any questions so far? Good. Okay, so as I mentioned at the beginning, this superdiffusive movement has been observed in biological experiments. So, for example, here on the left, we have the mean square displacement of 12 individual cells in a medium without chemical. And what we observe here is that for some runtime tau, the trajectories of these cells lie between normal diffusion, with slope 1, so this is a classical random walk, and strain-like motion with slope 2. This is what I called before a ballistic. Okay, so my cells are moving faster than just normal diffusion, which means they are doing a superdiffusive type of movement. In the second example, we have the distribution of the run and tumble time intervals for the case of the bacteria E. coli. And here the gray line is the tumble times and the black line is the run times. As we see here, these tumble times are distributed according again to this power law. So it's a heavy tail distribution, which again evidences this type of non-local diffusive movement. Okay? So, first I'm going to describe what happens at the individual level. So I'm going to describe very simple rules that my bacteria is going to follow, and then I'm going to take a limit to obtain a macroscopic description of this microscopic movement. Okay, so I'm going to consider an individual in a medium in Rn, so no boundary conditions, with some attractant concentration ρ. As I said previously, this individual is going to run for some time tau, which is distributed according to this probability psi. Here S is a function of the local concentration ρ of the chemical, and also on the total gradient of the concentration along a run, which is given by this expression here. Again, V is a velocity which takes values in the unit sphere, and alpha is a parameter that is between 1 and 2. Okay, so after some time tau, then the particle stops with the frequency given by beta, which again is expressed in terms of this running probability psi. And then once the particle stops, it can choose a new direction, V prime, which is symmetrically distributed with respect to the previous direction, according to this turn angle distribution case, which is also normalized. Okay, so we have just described for how long the particle is going to move, with what frequency is going to stop, and how it's going to choose the new direction. So three very simple rules. So the general strategy to obtain this type of macroscopic equations starts with this system of transport scattering equations, where F is my mesoscopic density that depends on x, t, v, and also the runtime tau. So the left-hand side here describes the run phase of the movement, while the right-hand side gives the density of individuals that are stopping with the frequency beta. Okay? These individuals that stop, they immediately start a new run at tau equals to 0 in a new direction v prime, which is described by this second equation. Then, of course, since I want to go from a micro to a macroscopic description, I have to scale in space and time, very similar to what Tommaso did before, with some peculiarities, because now we are in a non-local scenario. So here we scale on the variables. Here mu is greater than 0, gamma is a parameter that is between 0 and 1, and epsilon, it's a small parameter that is defined as the mean run time over the macroscopic time. So this is very small parameter. So since I want to get rid of all these microscopic quantities, the next step is just to integrate with respect to tau, and we obtain this kinetic equation, which you could think about as a Boltzmann type of equation, okay? Because here on the right-hand side, instead of having a gain-lose term coming from the interactions, what you have is something that is dependent on how cells change direction. But again, it's a gain-lose term somehow. So F, it's a new density that is independent of tau. Capital T, it's a third angle operator that basically describes how the cells change direction. And all the non-locality now it's encoded here in this convolution, in this blue term. So all this non-local movement that I've been talking about is described in this operator capital B, which we can explicitly write in the Laplace space. Now the next step is to simplify this convolution. And for that, what we are going to use is the quasi-static approximation. So we basically find the Laplace transform of this term, which is given by v epsilon hat, where lambda is the Laplace variable. And when we introduce the scaling, what we see is that as epsilon goes to 0, these terms goes to 0 faster. So we just get rid of this time dependency, and then the convolution is simplified just by using this quasi-static approximation. Then, with this simplification, we can go back to the main kinetic equation, where, well, I forgot to mention that this is a probability density function, which is related to this psi. So, all this term, you can write it explicitly, okay? Because previously you defined your psi function when I started just describing the individual movement. So this right-hand side has an explicit expression, which is given here, where we already see some term that resembles a kinetic term, right? So it depends on the concentration rho and the concentration of the chemical rho. And also we see something that resembles a fractional gradient. Okay, and then just to clarify, the type of definition of fractional gradient that we are going to use is given here. Okay, so it's very different from the one I showed you at the beginning, because this one depends on the velocity, which is precisely what we need. Okay, so now the next step, which I'm going to skip, is just to plug this into the right-hand side and compute the powers of epsilon. Okay. Then what we do is to obtain a conservation equation just by integrating with respect to all velocities. The right-hand side is just equal to 0 because it's the third angle operator is normalized to 1. So what we obtain is this conservation equation here, where U is the macroscopic density that now is going to depend only on x and t, and omega is the mean direction of the movement. Okay. So just to close this this equation here, what we have to do is to obtain the mean direction in terms of the density U. And for that, we go back to the kinetic equation. We multiply by v, we integrate with respect to all velocities, and then doing some standard, let's say, computations, we obtain this fractional chemotactic equation in the limit. Yes.
Sorry, you haven't set epsilon to 0 yet, right?
Yes, when you do this, you have to choose the appropriate scaling, so that's what I mean by letting epsilon to 0.
So you choose the scaling so that little w doesn't go to 0 when you let epsilon go to 0, is it so?
Yeah, exactly. Okay. So you see that here you have gamma, yeah. So this, if I go back, so here you have this, okay, so you choose your mean directions in such a way, well, the scaling of your mean direction in such a way that when you find the conservation equation, you get just epsilon, epsilon, and then 0 because this integrates to 0.
Perfect, thanks.
So this is what we obtain in the limit by choosing the appropriate scaling. And well, here the diffusion coefficient C alpha can be explicitly determined from the microscopic quantities. So in case you want to use this equation for a specific experiment, biological experiment, this is something that you can compute. And also, I want to highlight two more things. So the first one is that for alpha equals to 2, we recover the classical case of the Patlak-Keller-Segel equations. Okay? And the second thing is that this alpha parameter, which is related to the non-local movement of the individuals, only appears in the diffusion part of the equation. So the non-local movement of the particles doesn't affect at all the chemotactic component of the movement. And this was something that before it was not very known and very clear. So one of the advantages of having a microscopic description of this individual movement is that we can analytically obtain biologically relevant quantities, right? So we don't have to look at the actual particle movement. We can just draw some conclusions, some analytical conclusions from the PDE level. Okay, I'm not going to have time to discuss this, but this is also something that we consider in this work. Using more or less the same approach, we also studied the switch in cell migration, switching from superdiffusive to normal diffusive and vice versa. And here, well, we have two different types of cells, so we have metastatic and non-metastatics. And what was observed in some experiments was that for the case of the metastatic cells, the front and back movements are synchronized in space and time, so the cells travel long distances. And also these metastatic cells are known to invade the healthy tissue. So it was observed that basically in this case we had the metastatic cell that started almost like here, and it was traveling along the track. So this synchronized front and back movement just led the cell to travel very long distances. And what they observed was that the trajectories matched with the type of superdiffusion that I was describing before. On the other hand, for the other type of cells, so for the non-metastatic ones, what they observed is that the front and back movements are not synchronized, and what this leads is a very local movement of the cells. Okay, and let me see if I. No. No, it doesn't work. The previous one. Can I go to the previous one? Okay. So here at the bottom, you see this non-metastatic cell, which is almost not moving at all. Okay. So there is a big difference between these two, and this difference is given basically by this synchronization or non-synchronization between the front and back movements. So what we did in this work was just to consider a kinetic equation to describe this type of movement, where we included different levels of complexity, meaning that we described the movement either independently or with partial or full synchronization. Okay, and what we obtained in the limit was either diffusion, drift diffusion, or fractional diffusion, depending on this initial system. Okay, so actually we were able to compare some of the parameters obtained at the macroscopic level with the parameters obtained from the biological observations. Okay, so this was more or less the first part of my talk. Thank you very much. In the second part, as I said, what I'm going to do is I'm going to extend this individual behavior to a system of interacting particles. Okay, so here basically the main idea is to translate the loss of movement of biological system to the case of a swarm of robots. this was our main motivation at that time, but what I'm going to talk about here can be translated to any system of interacting particles, okay? It doesn't have to be robots, it was just the previous motivation. So we are going to consider a swarm of n individuals in medium, in a domain omega in R2 with reflective boundary conditions. Here, the diameter of each particle is given by rho, and again, this particle is characterized by its position, the runtime tau, and velocity v. For simplicity, we are going to consider that all robots or all particles are moving with the same speed c. And as I mentioned previously, in this case, we are going to have three main ingredients. So we are going to have non-local movement, alignment, and collision avoidance in this swarm. Okay, so the type of movement I'm going to describe is very similar to the run and tumble of the bacteria. So basically, we have that the robot or particle is going to move for some time tau, which again is distributed according to this power law distribution. Again, alpha is between 1 and 2, and this is even simpler than before because we don't have any chemical in the medium. Okay, it's just random movement. The robots or the particles are going to interact elastically, so momentum and energy are preserved, and there's going to be specular reflections. Okay, so the new velocity after collision is given by vi prime, which is given by this expression here, where nu is just the normal vector at the point of collision. Again, we have some stopping frequency given by beta, which again can be written in terms of the running probability. Now, when the particle stops, it can do two different things. So, with probability z, the robot or the particle can choose a new direction, vi star, according to the third angle distribution k. Okay, this is again this type of run and tumble movement that I described for the case of the bacteria. Now, with probability 1 minus z, the robot can align with their neighbors according to this alignment distribution phi. Okay, so lambda is the mean direction at the point xi, and it's expressed in terms of this non-local flux. This j is bounded, smooth, and tends to 0 at infinity. And it's given by this expression here. So it's just the integral of the mean direction of the particles with respect to some influence kernel. Okay? So basically, when the particle stops, it can do two different things. It can either align with their neighbors, or it can choose a new direction according to a certain distribution. So, in this case, we studied two different limits, and I'm not going to get into the details of any of these terms here, but I'm going to highlight one thing. So, we studied the long-time dynamics and also the short times. For the long-time dynamics, we have that the macroscopic density and the mean direction satisfy this system of equations, where this is just a term that is coming from the collisions, right? Lambda is coming from the alignment, and this is something that describes the fractional movement, this non-local diffusion. On the other hand, for short-time dynamics, so at hyperbolic time scales, what we observe is that the movement can be described by this system of equations, right, where we only have u and lambda, the alignment. So this means that at long-time scales, what we observe is a competition between the alignment and the non-local movement. While at short-time scales, what we see is that the movement is completely dominated by alignment. We don't see any non-locality in the movement at short-time scales. And actually this it's a very classical result for the case of swarming particles or self-propelled swarming particles. So at short time scales, it doesn't matter if it's a run and tumble or if it's super diffusive or if it's normal diffusion, it will always be dominated by the alignment of the particles. Okay, so I'm just going to briefly describe how we obtain these equations. And all the derivation is very classical for interacting particle, for Boltzmann equation, if you like. So we start with this system describing the n-particle system, right? Where now F is a mesoscopic density for the, well, it's an n mesoscopic density. Here my domain is omega capital N, which is given by this expression here, where remember that rho was the diameter of the particle. And what we are going to consider is that the volume occupied by the particles is small compared to the whole domain. Okay, this means that then the collisions between two particles are a rare event. And basically this is going to allow us to truncate this hierarchy and neglect collisions between 3 or more particles. Okay, and that's precisely what we are going to do here. So we just consider a two individual system where now we have a two individual density just for particle 1 and particle 2, which evolves according to this kinetic equation. Okay, this is very similar to the case of one particle. Also, we have to include some boundary conditions for tau equals to 0, just as we did before. And this means that, well, this is just describing that when the particles stop, then they can either choose a new direction coming from this probability distribution, or they can align with their neighbors. Okay? Just as I mentioned before. So now the strategy is very similar to the previous case. So first we have to integrate with respect to this quantity tau, the runtime. And then we aim to write a kinetic equation for the one particle density. Okay, so basically what we do is we integrate out the second particle. And when we do that, we see that the density P, well, the mesoscopic density P satisfies this kinetic equation. This kinetic equation has a term that describes the alignment, also a term that describes the long-range movement, and this it's very familiar to us already, right? Because it was the same type of convolution that we saw before. And of course, we are going to have an interaction term, okay? That again will come from integrating particle 2 on this left-hand side. Here n-1 just describes the number of particles that particle 1 can collide with. Okay, and this is also very classical for Boltzmann type of collisions. Okay. Now, again, what we have to do is to introduce scaling. So I'm going to start with the parabolic scaling, so the long time behavior. And, well, again, it's very similar to before, but now we also consider that the diameter of the particles are very small, while the number of particles tend to infinity. Okay? This is also called the Boltzmann-Grad limit. And also it's very classical when we deal with this type of interaction terms. Now the next step is again to try to simplify this right-hand side, so try to simplify this convolution and also try to deal with this interaction term. So to deal with the interaction term, what we do is the molecular, well, what we use is the molecular chaos assumption. In the regime that we are studying this system of particles, the molecular chaos assumption basically says that the velocity of the particles are approximately independent from each other. Okay, so then we can write this two particle density, so we can factorize this two particle density as the product of this one particle density. And this actually simplifies a lot our life when we deal with this term here. Okay, so these two particle densities are now written in terms of one particle density. So for the case of the convolution for the non-local term, we again use this quasi-static approximation, just in the same way that we did before, and even simpler because we don't have any chemoattractant in the medium. And then again, it's very easy to obtain a conservation equation, okay, just by integrating with respect to all velocities, and well, doing some tedious computations, we go back to the kinetic equation, and you obtain an expression for the mean direction W. And then what you see is that in the limit, this is the system of equations that describe this alignment non-local movement. Okay, so just very briefly, I'm going to explain what happens at short time behavior, so at short time scales. For this case, we introduce a hyperbolic scaling. So basically what we do is we scale space and time in the same way. For the hyperbolic scaling, we don't consider interactions between individuals. Okay, we simplify because it's already quite complicated. So in this case, what we note is that since space and time are scaled in the same way, we cannot use a quasi-static approximation anymore. So we have to find another way of dealing with this convolution term here. And what we do is we obtain a generalized Chapman-Enskog expansion in this way, where here we have some alignment terms. Okay. So then, well, I'm not going to give the details, but then with this expansion and the kinetic equation, we can obtain this system in the limit. Okay, it's very standard for swarming particle systems, the procedure to obtain this type of macroscopic equations. Okay, so as I mentioned at the beginning, this was motivated by a swarm of robots, and we were able to compare our results coming from the PDE to the robotic experiments. So in this case, of course, we are going to consider very simplified equations, so just a fractional diffusion equation. Well, of course, we have the size of the domain, which now corresponds to the size of the arena where the robots are going to move. We know the diameter of the robots and also the speed of the robots. And these other parameters are computed from these other parameters that we are going to need for the PDE. The initial condition for this experiment is just you have an arena and you place the robots in the middle. Okay, and it's the same type of initial condition for the PDE. Of course, here we only have 5 robots, but we also did the experiments for 10, 15, and 20 robots. Okay, and in all cases, they were placed, like a blob in the middle, just a ring facing away from each other. And what we did, we investigated area coverage and heating times. So here I'm going to just talk about area coverage. For different number of robots and different values of the alpha. For the macroscopic PDE, this is the way that we define the area coverage. So basically when U, so the macroscopic density, is bigger than 1 over omega, you count the area as covered. And when U is less than 1 over omega, then the area is covered with certain probability. For the case of the robotic simulations, it's very easy to just count the number of cells visited over the total number of cells, and that's your area. So here at the top, we have a comparison between the macroscopic simulation, which is given by this black line, and also the average robotic simulations is the red line. And this was for the case of 20 robots. with alpha equals to 1.5. And what we observe is a very good agreement between both solutions, especially at long times, okay? Because at long times is when the PDE is actually valid, okay? Also, at short times, we have a very strong presence of interactions, which for these simulations, we are avoiding these interaction terms. So here, it's well, this difference is also driven by the simplification of the model. At the bottom, what we have is a comparison of, again, area coverage at the final time, but in this case is for different values of the alpha. Again, the red curve is, corresponds to the robotic simulations, and the black line corresponds to the microscopic simulations from the PDE. Here what we observe is that as we decrease the value of alpha, so as the process becomes more superdiffusive, then the area covered is bigger. Okay, so this gives a very good strategy to cover an area. So if you have a bunch of robots and you want to think about which is the best strategy to cover a certain area, so well then try to do a superdiffusive movement. Okay, and this also was for the case of 20 robots. In this work, we also we also studied heating times. Okay, so it's the time that it takes for a robot to find a distant target. And again, we use the PDE to compare with the robotic simulations. Okay, so just to finish, I would like to mention some related work. So, concerning the first part of my talk, we also looked at space-time fractional diffusion equations for the case of T cells in the brain. In this case, we consider non-local movement and also long waiting times. Okay, this is translated into fractional time derivative and also, as we saw, a fractional space derivative. Related to the second part, we did this together with Tommaso, where we studied the switch in T-cell migration, but this case was driven by binary interactions. Okay, so the previous case I showed you was driven by mechanical properties of the cells, while here it's just by interactions between different species of cells. Again, the strategy was kind of similar, so we started from a kinetic equation that described these different particles dynamics. So the first term just describes the random movement of the cells, and the second term describes the interaction between different species. And what we studied was how these different interactions could lead to different behaviors in the cell. And finally, also related to the last part of the talk, we studied fundamental derivations of follower-leader models together with Sara Bernardi and Kevin Painter. And in this case, we had two different populations. Followers and leaders were fixed populations, not as in the case of Tommaso, where they could change the phenotype. Here we focused on the case of a swarm of bees, so they are very defined who is the leader and who is the follower. And then again, what we did was derive what kind of dynamics appear when you have these two different populations, and how many leaders do you need to keep the swarm moving and things like that. Okay, I think that's all from my side, so thank you very much.